Hello, in this video we will discuss about the real-time PCR for corona infection detection in infected person. So for example, a person which is infected. So first of all we will check their sign and symptom. After the checking of the sign and symptom, we will take the sample through a nasopharyngeal swab or oropharyngeal swab. So after this, taking the sample will store into the specific temperature. So after the storage of the sample, basically it is used for the several purpose for the PCR, amplification of the gene for detection of the coronavirus infection, post, pre, as well as infection time. So here let's begin to understand sign and symptom for detection of the coronavirus with the physically, the coughing, the dry breathing, and the dry breathing, coughing and sneezing, the fever, fatigue and short breath, as well as the fever will temperature will high due to the coronavirus infection. So in this way, take sample and reverse transcription of viral RNA to complementary DNA for application of viral genome into the PCR. So the polymerase chain reaction after the reverse transcription to become a complementary DNA, which that is contain basically the gene of the virus. So let's begin to understand first of all the virus structure. So here is the we will collect the data, collect the sample which contain virus or infected cells which that contain a viral RNA. So the viral RNA contain a spike protein ACE and RNA, which that is the ACE receptor are present in our lungs. And here is a reverse transcriptase enzyme also, and COVID-19 is here. You can see that it contain RNA. So let's begin to understand the principle of reverse transcription of PCR. So the first step is the reverse transcription because the RNA should be converted into the DNA, the complementary DNA for the further polymerase chain reaction. So the principle of reverse transcription of PCR is the same mechanism in which the reverse transcriptase enzyme synthesizes the complementary DNA from the RNA. The process has two steps. So the reverse transcription process and amplification and quantification will be occurred. That quantification is due to the we can quantify the severe infection or pre-infection and post-infection time. So in this way we will take the sample and use the sample, and this sample will be put into the microcentrifuge tube. After the putting into the microcentrifuge tube, the master mix will be added. Master mix can be reverse transcriptase enzyme, messenger RNA, buffer, the lysis buffer and elution buffer, and deoxynucleotide triphosphate, which that is the nucleotide we can add for the synthesis of the another strand to become a complementary DNA, and sequence specific primer will be added. That sequence specific primer is used and bind with a specific region of the gene of virus for annealing process to become an extension, and this extension will lead to produce a complementary DNA. And we can also put the DNA polymerase for the polymerization of the deoxynucleotide triphosphate into the reverse transcription to become a complementary DNA double helical structure. So this is the first step, vortex. After the vortex we will mix that things and washing. After the washing, that is the reverse transcriptase enzyme will be used, and this is the reverse transcriptase enzyme will complementary DNA formation, and in this way that is the after the washing and the complementary DNA after reverse transcription will be occur. So here is the complementary DNA after reverse transcription will produce the DNA, and this DNA is useful for the amplification of the gene for detection by a color, for amplification of the color for detection that gene if the color will be positive, so that is why the positive test. So here you can see this is the step 2. In this way the complementary DNA will be added to another tube. There are the multiple preparation of multiple well. Multiple well will be add the sample as a complementary DNA, and complementary DNA and DNA polymerase enzyme, buffer, and deoxy nucleotide triphosphate, and sequence specific primer and TAC-man probe will be added. DNA probe based real-time PCR for detection through TAC-man probe. So in this way here is we will add in the thermal cycle for amplification of gene. Let's begin to understand the DNA probe based real-time PCR TAC-man probe. So in this way here you can see this is a double-stranded DNA template that is come from the complementary DNA. This is come from the reverse transcription before the putting into the PCR. So in this way the 95 degrees Celsius temperature for denaturation of this complementary DNA into the single stranded due to the hydrogen bonding will be breakdown. So in this way the two strand 3' to 5' and other is the anti-parallel to produce. After this annealing stage, the 65% primer should be melting point 70 degrees Celsius. So that is why we can add the 62 degrees Celsius temperature for the annealing process, so that primer will be bind. Primer can be forward and reverse primer, and also the TAC-man polymerase enzyme is used for it, and TAC-man probe we will choose. What is the TAC-man probe? Here let's begin to understand. When light will be fall on the culture, so the culture will absorb the light and will not transmit the probe. So that means the DNA probe will be not producing light, but in this way the culture will be separate, so the light can be produced. So after this light producing mechanism we will discuss here. Just is the bind with the DNA probe, that is a specific, and binding with the probe with quencher. So the quencher will be released after the TAC-man polymerase or DNA polymerase enzyme. During the extension the 70 to 72 degrees Celsius temperature will required. So the quencher will be released and the color will produce due to the releasing of the quencher with the probe. So the probe will produce color after the light falling on this. So in this way let's begin to understand the amplification. So the amplification, the multiple copies of the genome will produce, and with a copy of the genome will also produce the color. So the intensity of the color will be increased with the increase of the amplification of the genome. So the endogenous 5' nucleases activity cleave the probe. So remember about this. So after this the color production, the analytical data report will be transmitted into the computer or machine or software for detection in the graph. So if the color will produce more, so the detection will be positive. If the color will not produce with a specific, it means the DNA of the virus will genome of the virus will not present, and in this way the viral detection will be negative. So here is I am drawing the graph for understanding the analytical data report in the computer machine, which that is show the color is the vertical as well as the cycle will begin through a horizontal time. So here is the threshold are present, which that is crossing the threshold mean the going to the positive, and the CQ value will be increased after the crossing the threshold, and is exponential phase with the color production, and the more color produced it means the positive test. And if the non-exponential phase will begin, in this way the color will not more produce, so we can done this process and complete. So let's begin to understand the real-time PCR mechanism for detection of color. So there are different color will produce, but our choice is the orange color, so it means the orange color will produce more, so that is why we can say that the coronavirus infection is positive. So here is the real-time PCR function we will discuss after the amplification of the DNA in the multiple valves. So we are preparing the multiple valves for amplification of the DNA, the amplification of the genome. So when more amplification of the genome will produce more color. So that is why when more color will produce, it means the coronavirus infection will be positive. So here is the tungsten halogen lamp we will add, and this tungsten halogen lamp is same like a sun light, and in this way the excitation filter we put in the front of the tungsten halogen lamp, and we will put also the mirror, and this mirror will transmit the light into the multiple valves, and after these multiple valves we will put that is the mirror also two mirrors, and one mirror is also emit the light from the color from the tube, test tube. So in this way the microcentrifuge tube is basically is the color will emit, and that color is emission of that color will be go into the mirror, second mirror. After the going into the second mirror, that will collect the data in the form of color from the emission of mirror. So this mirror will produce a specific color, and this specific color will be collected by the CCD camera. So the CCD camera is here, we can say that is collect the data after the lightning or specific color will be produced from the excitation mirror, which they reflect the light, and after this reflection of this light will be collected by a CCD camera, and after this CCD camera will put into the analytical real analytical data for checking the color, and the specific color will produce if it means the coronavirus infection will be positive. So anyhow, in this way we will discuss about the whole graph for understanding the pre-infections positive by PCR and post-infections positive by PCR and infection time. So here I am drawing the graph for understanding well. So this is the upper line shows that is the low analytical sensitivity, and it means the coronavirus infection will be positive. It means the easy to analyze, but the high analytical and sensitivity in the PCR should be after the infection as well as the pre-infection, and in this way because the RNA content will be less. So that is why. But in this case here you can see the positive test show the orange color, and this orange color is the here is the increase, and here is the pre-infections positive by PCR will be occur after the 7 to 12 days, and after the 12 days the infection will be severe and viral load will be increased, and in this way the viral load will lead to think about that just a viral load will be more, it means the infection time, while the post-infection positive by PCR. So here's the more cycle to produce more color. So it means the cycle require more after the post-infections positive by PCR because the genome will be less. So thanks for watching. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. Bye. Thanks.